Hello everyone, my name is Juan Martinez and today I'm going to be presenting a poster that along with my mentors Jyrko, Correa and Belarmino Gonzalez created throughout the summer semester in Miami Dade College campus. The poster is on the beta-entropy matrix distortions of the lattice of partitions. In this study we investigate the distortion among beta-entropy matrices, focusing particularly on how the parameter beta affects the distances between partitions. We compute the specific values of this matrix and distortion between them using beta equals 1/10 and beta equals 1/2. We applied our results to compute the distances between non-standard mitochondrial and nuclear genetic codes and use the proximity between the genetic codes to explain these distinguishing characteristic features on the evolution of organisms. The objective of this poster is to analyze the structural similarities and difference among distances between partitions using the beta entropy metric. The main focus is to highlight the deformation that the beta parametry does on the distance between partitions when it changes. First of all, I would like to highlight some core definitions. The first one being set X is a data set. In this case, we have that set X is equal to A, B, and C, meaning 3 elements. A partition of X is a collection of non-empty and disjoint sets whose union is X. In this case, we have P prime. And then we have the concept of clusters, any element of the partition of X. In this case, we have highlight over here, a cluster being A, B. Then we have the spatial organization, which is a hierarchical organization into n-1 levels, and where we have our set X partitioned in different sections. In the level 0, we have the partition M of X has 3 clusters, and by measuring those clusters, we can now get the level 1 and 2. The metric study on this poster was the beta entropy metric, and what it calculates is the entropy between 2 partitions P and P prime, and then the partitions from P prime to P, and then the addition between these entropies give us the distance between these partitions. And here in the calculations between the partition of the set X using 2 different betas, we have beta equals 1/10 and 1/2. We can see that here beta equals 1/10, the distances are greater than when we use beta 1/2. Then we have the concept of distortion. Distortion is the measurement of variation between different metrics. In this case, it's defined as the product of the contraction and expansion. And we can see here in this graph that when we have one parameter beta, one matrix fixed, and we compare it to other betas that are farther from this beta parameter or beta prime values, we can see that the distortion size increases as we put farther apart these beta parameters. And when beta is equal to 1, the equation is undefined. In the biological application of this topic, we can see that we can create a set X being 64 trinucleotide codons, the partition being genetic codes, and the cluster being grouped by 21 amino acids. And here we compare the distance between the standard genetic codes and other 16 created genetic codes. In this case, we can see that the group of mitochondrial codes are the ones that showcase the greatest distance between the standard code. And this might suggest that the mitochondrial genetic code had a different evolutionary pathway than the nuclear codes did. But in order to make such a conclusion, further investigation should be done about the topic. Now the conclusions. The beta parameter, beta and beta prime, as this parameter changes, the distances among the partitions vary. Consequently, the distortion also fluctuates. Additionally, the number of elements, if the number of elements X increases, also the distortion size increases. Finally, the beta entropy metrics can be used in order to calculate the difference between genetic codes. And some acknowledgements will be like this work was made possible by the support of the Eduardo J. Padron campus and the School of Science in Miami Dade College. The Summer research summarizing the following supported in part by the U.S. Department of Education Grant Award STEM Engine. Any opinions, findings, and conclusions of the recommendations expressed on this material are those of the author and do not necessarily reflect the views of the funding agency. Thank you so much for your attention. See you in the next one.